let's get started. Thank you for joining us here today, everyone. And yeah, I'm Ryan Crawford. I am a PhD student in Evan Snicken's lab. And in the Snicken lab, we use bioinformatics approaches to study the evolution and epidemiology of bacteria, and with specific interests into bacteria that affect human health. So a little bit of a background on bacterial genomes for those of you who are less familiar. Bacteria typically have a single circular haploid chromosome, although additional extrachromosomal DNA can be present in the form of plasmids. They differ really substantially in comparison to eukaryotic genomes in that they're much smaller. The organisms that we're going to be discussing today have a genome of about 5 megabases, and the proportion of coding sequences is much higher. And in contrast to eukaryotic genomes, there's really high correlation between the number of genes and genome size. And importantly, even within a species of bacteria, gene content can vary really substantially, which brings us to some important definitions that we're going to be covering throughout the talk. When you think about bacteria, there's this idea of a core genome in contrast with an accessory genome. So if you look at this Venn diagram where each of these ovals represents a genome of a species, the intersection of all of these genomes is referred to as the core genome. And this typically encodes essential gene families that are used for vital metabolic functions, for example, DNA replication machinery and genes involved in protein synthesis. However, this is just a fraction of the total number of genes that is present in each genome. And these variably present genes within a species are referred to as the accessory genome. And these are largely dispensable and may be gained and lost in response to various selective pressures that an organism may be experiencing in its environment. And the portion of the genome dedicated to the accessory genome can actually be much larger than that of the core genome for many species, although this phenomenon is variable depending on what species you're talking about. And the dominant source of how genes are gained into the accessory genome is called horizontal gene transfer. And this involves direct movement of genetic material between two cells. And so this phenomenon enables really rapid phenotypic evolution because organisms don't need to evolve these functions on their own. They can just inherit them from a donor into a recipient and then start expressing those genes and gaining the phenotype. And a very clinically important example of this is antibiotic resistance, where genes that metabolize antibiotics can be really rapidly spread within microbial communities. And so in the Snicken lab, we view these patterns of evolution from a phylogenetic perspective. So a phylogenetic tree is how we visualize the evolutionary relationship between organisms. So the branch lengths shown on the tree are directly proportional to how related the isolates are through a common ancestor. So we use phylogenetic trees to study important phenotypes, like I mentioned, antibiotic resistance or transmission of bacteria in healthcare settings, as an example. And for projects I was working on in the lab, I really wanted to generate phylogenetic trees for really large numbers of genomes in the range of tens of thousands. So existing methods for this didn't really accommodate all of the things we required with respect to reusable memory usage and runtime. Additionally, I wanted to avoid downloading terabytes upon terabytes of FASTQ files. So using genome assemblies is ideal. Additionally, this enables us to expand our sample size using genomes that are sequenced on different platforms, PacBio, Illumina, or Nanopore, for example. And additionally, I wanted to use as many phylogenetic markers as possible in order to build the alignment and tree. And for a lot of the organisms that we deal with, they are not very well studied. So shared marker genes for typical bacterial typing schema, which focuses on, 7, a handful of housekeeping genes, might not exist for all the species that we are dealing with. And traditional methods of this, like selecting, the 16S ribosomal rRNA, is a commonly used tool for these kind of comparisons, but it really has limitations with respect to how many variants you can observe between genomes, which is a major limitation because a lot of the genomes that we study are very closely related. So commonly used methods for generating alignments, which can then be used to build phylogenetic trees, as in human genetics, involve aligning reads to a reference genome. But selecting an appropriate reference genome for bacteria is a bit of a challenge because, as I said earlier, there can be so much genetic diversity within a species. Picking the best reference genome for your isolates is not an easy task. And additionally, even within a species, when there are very diverse genomes, this can perform really poorly because the scale of the genetic diversity amplifies the probability of mismatched reads. There have been previous methods which use K-mer and the min-hash algorithm in order to calculate genetic distances between isolates. And these are computationally efficient, but they can suffer from confounding due to the accessory genome. So isolates could share a plasmid, which is a few hundred kilobases, and that which might be nearly identical between two isolates. And that would make isolates look more closely related than they actually are. And so we wanted to kind of get the best of both worlds and getting an estimation of the genetic distance between organisms, but additionally, we want to produce an alignment which can be leveraged in the future to give insights into molecular evolution and downstream analysis. So to this aim, we developed Cognac, which stands for Core Gene Alignment Concatenation, which we recently published in BMC Bioinformatics. And to give an overview of how our algorithm works, we start with the input assembly and the annotations in the form of GFF files. So we use the coordinates within the GFF files to extract the coding sequences, and then we translate the coding sequences. So this effectively reduces the size of the sequences which need to be aligned by 1/3, which is important because length is a major contributor to complexity in multiple sequence alignment. And additionally, we can leverage the redundancy in the code-on-code and more accurately identify orthologous genes. So when you're looking over very large phylogenetic distances, especially, the nucleotide sequence isn't very well maintained, but the coding sequence itself is well conserved. So it's easier to identify the orthologous when using the amino acid sequence. And so we use a program called CD-HIT to identify our orthologous genes. And what this does is uses an algorithm to cluster the amino acid sequences by the identity, and we provide a length threshold so that we have approximately the same amount of aligned sequence, and make sure that, you know, at least to a certain degree, a given gene is still has the same functional capacity. And then we parse the CD-HIT results and identify the core genes from that. And by default, our algorithm defines core genes as present in 99% of genomes. This is important because it allows for some degree of errors that can occur when building an assembly, or errors in gene annotation. And we also ensure that it's present as a single copy in 99% of genomes. So there can't be paralogs evolving independently of each other, amplifying genetic variation. And additionally, we make sure that there's at least one variant. It wouldn't be a phylogenetic marker if the gene had no variation in it. So we then align each gene individually. And so for this, we also want to minimize the number of sequences which are included in the alignment, because the number of sequences grows exponentially with each sequence included. So first, we identify the unique alleles, which we defined as exact string matches. So the idea here is that finding identical sequences is computationally efficient, but multiple sequence alignment is very complex. So if the red sequence and the purple sequence are identical, then we would only align the red sequence, and then we do the alignment with mapped and parse the alignment so that we duplicate the aligned sequence for any alleles which were duplicated. So generating the alignment for the full set of genomes. And then we do this in parallel, enable multi-threading, so this can be performed on as many cores as are available. And so next, the individual gene alignments are concatenated. And we also enabled several optional output files, including the ability to reverse translate the amino acid alignment back to DNA. So the idea here is that you get all of the computational benefits with respect to efficiency that are involved in generating the amino acid alignment, but you can amplify the amount of variation produced by redundancy in the codon code to distinguish closely related isolates, producing an amino or a codon-aware nucleotide alignment. Additionally, in our R package, we included functions for generating distance matrices, and from those distance matrices, you can create neighbor joining trees with the 8 package, and all of that is included as well. So to benchmark, we selected 8 unique genera of clinically relevant bacteria. These represent human pathogens, which are commonly associated with antibiotic resistance. And there were various numbers of genomes available. We used the Patrick database in order to collect publicly available genomes, which are of good quality or complete. And they curate this database so the genomes which are included have reasonable degrees of contiguity as well as presence of essential gene families. And there were there's a high degree of variability between the number of genomes that are available. Escherichia, for example, which includes E. coli, which includes human commensal bacteria, as well as pathogenic variants. There were nearly 12,000 genomes available. And then for genomes which are genera that are less studied, Pluralibacter, for example, there were only 24 genomes available. So first, we look at runtime, shown here in minutes on the y-axis, and then the number of genomes is on the x-axis. We were very happy that our algorithm finished in less than a day, even for these really large data sets with greater than 10,000 genomes. And runtime scaled linearly as the number of genomes increased. We then wanted to look at runtime as a function of each step. So in these bar plots, it shows the runtime for each genus. Klebsiella, again, took the longest as a fraction of the total runtime. Aligning genes was the longest step for every run that we did, with the exception of Pseudomonas, where finding the orthologous genes had the higher fraction of time devoted to it. This is because Pseudomonas has a larger genome size and greater accessory genome variation within the individual genomes. Next, we wanted to look at how much effort was saved by only aligning the unique alleles for each gene. So in these bar plots, each position along the x-axis is an individual gene, and the y-axis is the number of unique alleles as a fraction of the number of genomes. So if every genome had a unique allele for that gene, it would be 1. If there were no unique alleles, it's 0. So, sorry, there's a dog barking in the background. At the high end, Pluralibacter had a single gene which had 80% unique alleles, but when you take the median value, so the middle of each of these curves, and then plot it against the number of genomes, you can observe this really stark inverse relationship where E. coli and Klebsiella each only have about 1% of the alleles being unique relative to the number of genomes. So you're really saving yourself a lot of effort by not aligning all of those redundant sequences. And it just shows that because bacteria are related clonally, adding an additional genome really doesn't increase the amount of sequence space to a high degree at all. So I next want to illustrate a couple of projects that we've used this for in the lab. So the first project we applied our algorithm to was to analyze the distribution of capsule genes in the Bacteroides phylum. So bacteria can have a capsule made of complex carbohydrates surrounding the outside of the cell. And these can vary depending on the specific bacteria, and individual bacteria can produce a number of different carbohydrates. So we worked with the Martins lab to show the distribution and the number of loci which encode these capsular polysaccharides throughout the phylum Bacteroides. So even over this incredibly large phylogenetic distance of phylum, we were able to find conserved core genes and then make this maximum likelihood tree. Additionally, we have scaled up a bit since then, and we now have assembled a data set of nearly 16,000 genomes. And even at this really high level, our run finishes in less than 24 hours. So now in future projects, we are using this data set to integrate how antibiotic resistance genes spread across geographic regions and have evolved over time. And so, as I mentioned earlier, we not only wanted to get an idea of genetic distances, but we also wanted to produce an alignment that can be used further to study molecular evolution. So our first case study of this was another collaboration with Eric Martin's lab, and they were interested in how human gut bacteria can diversify their phenotypes in response to changing environments and the various kinds of metabolites which they can use as a food source. So if you are unfamiliar with the microbiome as a whole, there are symbiotic bacteria that live in your gut, and these are actually responsible for a majority of the digestion of metabolites generally, but specifically complex carbohydrates are of interest here. And they had measured the ability of a data set of over 350 bacteria to grow on 45 different metabolites. And they noticed that very closely related bacteria can have really different metabolic profiles with respect to their metabolism. So they came to us to see if we could identify specific genomic events that may have helped shape their metabolism and how these events were happening. So one major way that horizontal gene transfer happens in bacteria is recombination of donor sequences into loci in a recipient genome. So if we see this donor sequence in yellow and the recipient sequence shown in red, they can share common genes which facilitate recombination into that locus. And additionally, there might be other genes involved in this horizontal gene transfer event, which then expand the pan-genome. So new metabolic functions may arise due to events such as this. And so we wanted to look at a data set of genomes and see if we could identify any loci which exhibited this pattern of variation. So a brief overview of our pipeline: we first identified core genes with cognac and created the concatenated gene alignment. We then partitioned that alignment and built the individual gene trees. And then we wanted to identify specific gene trees that exhibit patterns of horizontal gene transfer, and then see if there were any loci where there had been pan-genome duplication events at a locus that exhibited patterns of HDT. So to do this, we collected a data set of Bacteroides xylanosolvens and ovatus. And this is maximum likelihood trees shown here. And you can see that the two species exhibit clear clustering on the tree, with xylanosolvens shown in red and ovatus shown in blue. And if individual genes were all evolving independently of each other, the individual gene trees would share the same pattern of variation and reflect the species tree. But there are many instances where this is not the case. So we identified these with a simple approach of, for each individual allele of a gene, we would calculate the distance to all of the members of the same species and then take the median value there, and then do the same for all the members of the opposite species, take the median value, and then we can plot these out in 2 dimensions to show any instances where the distance to the same species is higher than expected, and the distance to the opposite species is lower than expected. So each point in this figure represents an allele of a gene, and the distance, or the median distance, to the ovatus alleles is shown on the y-axis and Xylana solvens on the x-axis. And as you can see, the lower and upper half of the plots are mostly blue and red, respectively, showing that typically genes have a closer identity to the same species relative to the opposite. However, there are many cases where this is not the case. So we kind of picked off the genes that are obviously exhibiting patterns, which may indicate horizontal gene transfer, as above a threshold of a distance threshold of 0.1. And then we looked at the specific coordinates that these genes occur on the chromosome to find cases where the locus shared the same composition with the genes in the exact same order. So perfectly syntenic and collinear between genomes of the opposite species. And then once we identified these loci, we could find genes that do not occur in absence of evidence of HGT. So one such locus was of particular interest. This figure is a little bit complicated, so let me walk you through it step by step. Each column in this heat map represents a genome, and the genomes are sorted by the tip labels of the phylogenetic tree I showed earlier. Again, clear clustering with the Xylana solvens on the left-hand side and ovatus on the right. And then each row in this heat map represents a gene. And the colors indicate various types of genes. Core genes are shown in shades of red to blue. The alleles that in core genes, which are more similar to Xylana solvens, are shown in brighter shades of red, and more like the ovatus are shown in darker shades of blue. Genes which are absent in a given genome are in light gray, and genes which are present are shown in dark gray. And HGT genes, which only occur in species with evidence of HGT in the core genes, are shown in light blue. And the order of the rows here is reflective of the order of the genes on the chromosome in which the HGT event occurred. However, genes can be present at different loci. So in every genome, this order is not maintained. However, genes can still be present at a different locus. So what we observed here is a horizontal gene transfer, which we hypothesized came from the ancestor of the Xylana solvens into ovatus. So not only was there one gene which we identified as being likely transferred between these two species, but several of them which all line up all across the chromosome. And additionally, these accessory genes here, which are which is a locus which degrades a complex polysaccharide called mannans, which is present in the cell wall of grapes, for example, appears to have been donated along with these HTT genes. However, it seems to have been lost in some of these Opatus strains. And our collaborator, Eric, was really happy to see this because they had actually done genetics on mannan degradation in Ovatus previously, and we're really interested to see that we could actually identify horizontal gene transfer between these two species as a source of that phenotype. So conclusions. We have developed cognac, which is a robust and rapid method for generating concatenated gene alignments. And we were able to build it in a way which enables it to scale to extremely large data sets and still maintain reasonable memory usage and runtime. And additionally developed a data-driven approach for finding phylogenetic markers that are common to a given data set. And we were able to use several algorithmic tricks which enable us to minimize which enable us to maintain efficiency. And we're using this in the lab for many different applications. Yeah, hopefully useful in the future. And additionally, we can use these alignments to study molecular evolution and use the sequence variation in the concatenated gene alignment to identify HTT loci and apply this to a data set of xylanosolvents and Ovatus genomes. So for future directions, I want to package up the functions that I wrote for the HTT project as an R package. And a weakness of the setup as it currently exists is that it's really just built to compare two distance matrices with the distance for each allele in each genome and just do those two species comparisons. However, this is really an underrepresentation. There are multitude of sequence species of bacteria. So it would be ideal to set this up where you could do multiple species comparisons and find minimum. So you could set it up similarly to a tensor where you have genes, genomes, and species, and then you can imagine looking through the tensor and seeing areas where there are minimums, which might correspond to horizontal transfer between two species or two different phylogroups via sequence types within the same species. And ultimately, I'd like to apply this to study the natural evolution or the natural history of epidemic lineages of bacteria. Specifically, we're interested in Klebsiella pneumoniae, ST258, which was first identified in the 90s and has since spread across the globe and is strongly associated with antibiotic resistance and morbidity and mortality globally. And I ideally would like to use this to look at the genomes, how they've evolved over time, and identify loci which have high rates of genome transfer or potentially are maintained as the organism is spread globally. And are there any patterns of variation? Are there specific species which share genes more often or appear to have an origin from a second species and have been maintained over time? So I'd like to thank the SNCN lab. Everyone is great, and it's a great place to work. Also the Martins lab, my collaborators on the HGT project, Eric and Nick, as well as my committee. I'd like to thank the NIH and the National Institute of Allergy and Infectious Disease for funding, as well as the University of Michigan Bioinformatics training grant. And with that, I can take any questions.
If anyone has any questions, you can feel free to either unmute yourself and ask, or put them in the chat box and we can answer them from there. All right, oh, looks like a question just came in. So Ryan, I will read this to you. So your benchmarking included human-related species. Any predictions of looking more at environmental species?
Yeah, we haven't applied these to any kind of environmental isolates, but I would imagine that it would work just as well. There are, obviously fundamental genes which are found across these human species, as well as Pseudomonas. So that's incredibly large phylogenetic distances. We can still find shared core genes there. So yeah, I would imagine gene annotation algorithms would perform well on these, at least in finding core genes. There might be additional functions there which are not as well annotated, but yeah, it should perform on those kind of data sets as well.
Great, Ryan. There's another question that came in chat, so I'll read that one too. It says, "Hi Ryan, excellent talk. Wondering if you have predictions about using this tool set on actinomycetes. I know them to have very complex genomes and interesting HGT events."
I'm not familiar with those isolates actually. So yeah, they're
apparently soil bacteria.
Soil bacteria. Okay. Yeah, I mean, like I said, I would imagine it to be generalizable to those kind of bacteria, and yeah, it would be very interesting to see what kind of functions are horizontally transferred in that environment as well.
Does anyone else have any other questions?
Hi, I have a question. Hello,
Yeah,
am I audible?
Yep.
Hi, Ryan. So you showed the runtime slide, right? Where you show a linear scaling with genome sizes.
Yep, that's right.
So so apart from CD hit, what all softwares did you use to get those graphs?
So the only dependencies there are CD hit, and then MAFT performs the alignments.
Okay.
So yeah, just those two dependencies.
Okay, and what sequencing technology did you use on your input data set? Was it Illumina, Nanopore?
So it's a mixture of all of them.
Okay, I see.
Yeah.
Okay, and one last question. So did you face any noteworthy system memory issues, like depending on the compute machines that you worked on?
So yeah, I've run this on my computer, which runs Ubuntu, as well as Great Lakes, which is CentOS, and nope, haven't had any problems yet. But yeah, I haven't tested it on a Linux or on a Mac environment or anything like that. So All I
Okay.
can say is there have been no problems on Linux so far.
Okay, and your Linux was a laptop, right? Or
Yep.
okay, okay. Thank you so much.
Yep.
So maybe we'll just give it another minute or two in case anyone else has questions. Again, please feel free to either unmute yourself and ask your question or put it in the chat box and I can read it to Ryan. All right, well, I don't see or hear any other questions, so I would like to thank Ryan for giving such a great presentation today and taking the time. And I'd like to thank everyone who attended for joining us today, and hopefully I will see everyone here next week when we will learn about FSeq2, improving the feature density-based peak color with dynamic statistics. So thanks, everyone.